Our first research highlight is titled Integrative Analysis of Gut Microbiota, Microbial Gene Expression and Function coupled with Metabolome to Understand Mechanisms that Influence the Development of Non-Alcoholic Fatty Liver Disease by Ekaterina Smirnova at Virginia Commonwealth University in the USA.
Microbiome plays an important role in non-alcoholic fatty liver disease. However, it has mostly been studied in isolation, which limits the translational potential of this research. Intestinal microbiome is involved in conversion process of specific group of metabolites called primary bile acids into the secondary bile acids. This conversion process happens when primary bile acids in green undergo bile salt hydrolase action, then enter the bacterial cell and undergo a multi-stage conversion process by bacterial genes, by A, B, F, N, and so on, and come out as secondary bile acids. If you would like to hear more about how we relate the expression of microbiome metabolites and their changes to bacterial genes, their function, to development of non-alcoholic fatty liver disease, I will be available for questions 11:50 a.m. New York time zone. Thank you.
Our next research highlight is titled Phage Induction Gives Insight into the Anaerobic Digestion Metavirome by Alessandro Rossi at the University of Padova in Italy.
In anaerobic digestion, a community of bacteria and archaea produces biogas. Of this community, the virome is the least known part. For this reason, I set up an experiment consisting of several batches undergoing different stresses in order to induce prophages. Viral and microbial communities were physically separated by filtration and sequenced separately. The following bioinformatic analysis reveals the viral community dominated by the three big families of tailed bacteriophages: Sifoviridae, Podoviridae, and Myoviridae. A fraction of genomes has been classified as Bicaudoviridae, which infect archaea. Among the most interesting findings is the presence of a great number of sialidases associated with tail proteins. They are involved in host interaction and in the destruction of the cell wall of the host, allowing the infection to occur. Another interesting hypothesis is the presence of a kill-the-winner population dynamics. To know more, join me in my dedicated room and follow our group at Bioinformatics on Instagram. Thank you.
Our next research highlight is titled Human Lung Microbiota-Driven Innate Immune Landscape by Sudip Das at DMF, University of Lausanne in Switzerland.
The human deep lung is one of the most immunologically active areas and is not sterile but has a microbiota. This microbiota originates from the oral cavity and ends up in the lung by microaspiration. In a recent study, we showed that there are 4 pneumotypes and the balanced microbiota is good for us. We have both the good and the bad bacteria from human lung in culture. However, we still didn't know the direct interactions with alveolar macrophages due to the lack of an in vitro model. So we decided to make it by differentiating monocytes with some culture tricks, and voila, we have alveolar-like macrophages. Using this, I chart out the innate immune landscape triggered by various resident lung bacteria, which showed different inflammatory potential, and some can protect against pathogens.
The next research highlight is titled The Aging Mouse Microbiome Has Obesogenic Characteristics by Dana Binyamin of Bar Ilan University in Israel. And unfortunately, we don't have an elevator pitch available for this presentation. The next research highlight is titled The Gut Microbiota's Influence in the Development of Fetal Alcohol Spectrum by Natasha Kitchin at Stellenbosch University in South Africa.
Hi, I'm Natasha, and today I'll be sharing some of our work on the gut microbiome in fetal alcohol spectrum disorders. Fetal alcohol spectrum disorders describes 4 diagnoses along a spectrum of increasing severity consequential to prenatal alcohol exposure. South Africa has the highest prevalence of FASD worldwide. We found that ruminococcus was lower in women with infants diagnosed with FASD. while Alloprevotella was higher in these women. Unexpectedly, bifidobacteria was higher in infants diagnosed with FASD. A lower relative abundance of bifidobacteria has been observed in children with autism, making this finding unexpected. Prevotella was higher in infants diagnosed with FASD, a finding that mirrors findings in individuals diagnosed with autism in other low and middle income countries. These microbial differences may contribute to the neurocognitive deficits characteristic of FASD. Thank you.
And our final research highlight is titled When 16S Ribosomal RNA Outperforms Whole Metagenome Shotgun Metagenomics by Stanislav Smatana at Mariskova University in the Czech Republic.
16S rRNA-based taxonomy has many problems. Among them are the multi-copy nature of this gene, its alleged inability to identify species, PCR bias, and within-species sequence variation. Many authors have suggested that we should adopt whole metagenome sequencing instead. However, whole metagenome sequencing leads to waste of sequencing potential on non-specific areas of genomes. In light of this, should we use 16S rRNA or not? Well, we think that this is not the right question. We have conducted an extensive in silico analysis of tens of thousands of bacterial and archaeal genomes with thousands of primer combinations, and we have found that 16S rRNA can in many cases identify more species than whole metagenome sequencing at a lower price. In conclusion, you should feel free to use 16S rRNA. However, you have to be careful to choose your primer wisely. Thank you for your attention, and if you are interested in more details, you are more than welcome to join us at our research highlight.